Thanks, everyone. I'll be quick because I know, Andy, you've got a very robust presentation. So this is mostly for the fellows and trainees. The rest of us all know Andy quite well due to his sort of trajectory since he's come to Mount Sinai, which has been fairly impressive. So Andy Stewart, although he is an internal speaker, likely could have been one of our invited external speakers for multiple of numbers of our talks. I got to know Andy back in 2012 when he joined the Sinai faculty after coming here from UPitt, where he had been the chief of endocrinology and had a very robust career there. Prior to that, Andy had been at Yale for both his endocrinology fellowship and then stayed on as faculty. So why is Andy speaking about diabetes? So Andy is someone who is a clinician, not an MD PhD, who moved up the field in the area first of parathyroid disease. And then while he was doing his research, he noticed that PTHRP drives beta cell replication, survival, and expansion. And that led Andy to pivot into the area of diabetes, which of course gives me joy because, as everyone knows, I think everything really is all about diabetes regardless. Andy has 6 NIH active grants right now, and they're not ones that expire in the next year. So he's well funded and well supported by multiple different institutions in the past and presently. Truly impressive. And then just as a general comment, Andy's just been a nice guy. He's been a supportive mentor to me throughout my transition from clinical to research career. And he's just no nonsense and really works well with everyone. It's been a pleasure to introduce you, Andy, and I look forward to your exciting talk.
All right. Thanks, Carol. So, Emily, am I okay to upload?
Yes. Yeah, go ahead.
All right. Let me do this. Let me get a laser pointer here.
Perfect. Yep.
Okay, so thanks, Carol, and thanks everybody for coming. This is really a Mount Sinai story, and it's reached a point. I mean, you all have heard bits and snatches of this over the years, but I don't think I've spoken for a couple of years, and a lot has happened in the last couple of years. So the title of this is Human Beta Cell Regeneration for Diabetes, a Journey from Impossible to Possible. And you'll see where this is going. It starts off a little clinical. It gets a little basic sciencey, but not that basic sciencey, so hang with it. Then it becomes very clinical again at the end. So I'm going to give you a glimpse of what I think the future looks like. So I do have disclosures. Mount Sinai has filed patents on some of the work that I've been that I'll present. So I want to just begin with what we know about human beta cell mass proliferation and the discovery of DYRK1A inhibitors. So we all know that beta cell mass is reduced in type 1 diabetes. What's new about this slide this century is that this number is not zero. Most people with type 1 diabetes, even if they've had it for, Joslin medalists for 50 years or 60, 80 years, at autopsy, they still almost always have at least a few beta cells left. And this number is a moving target, depends on the study. Some of them say it's 50%, some say it's 2%. But the point is, everybody with type 1 diabetes still has residual beta cells, no matter how long they've had type 1. The other half of the story is that type 2 diabetes is also associated with a dramatic reduction in beta cell mass. So if you're obese and non-diabetic, beta cell mass looks like this. If you're obese and type 2 diabetic, beta cell mass at autopsy looks like this. The same paradigm follows for people who are lean, non-diabetic or type 2 diabetic. So the point of this slide is that in the world, there are 463 million people with diabetes, and they all need more beta cells. And so it begs the question of, what can we do to make beta cells regenerate? So what do we know about beta cell proliferation? We know that on the y-axis, this is beta cell proliferation measured by Ki67 labeling in autopsy studies. And then this is age in human beings from embryonic life to first year of life to childhood, adolescence, and adulthood. And so what you can see is that beta cell replication does occur in human beta cells, but only in the first year of life, and then it tapers off during childhood and adolescence. And so by the time you get to be a full adult, I guess my grandkids would argue that they're full adults at 12, but in any case, by the time you get to be an adult by our standards, the beta cell replication has ceased, and no one's ever been able to make it replicate. So that's a problem because we do get, and I'll show you, human islets from NIH for research. That's the good news. The bad news is they don't replicate, and no one's ever been able to make them replicate. So we're studying islets by definition, or beta cells by definition, that won't replicate. So that's a problem for us. Also, if you want to do clinical trials, if you do discover drugs, you're not going to be able to do them in 2-year-olds. You're going to have to do them in 50-year-olds. So it's a daunting challenge, and no one's ever been able to make human beta cells replicate. So we did a high throughput screen that we published in 2015. We, in particular, is Peng Wang, who's a senior member in my lab. And the bottom line is this: we screened using robotic high throughput techniques 100,000 compounds, and out of that we found one that's able to induce human beta cells to replicate. So the compound, and you guys have heard this before, is called Harmine. Harmine looks like this. It's a low molecular weight small molecule the size of aspirin or Tylenol or something. And its target is this kinase called DYRK1A. And Harmine fits into the pocket in DYRK1A where ATP fits. DYRK1A is a kinase. It needs to have phosphate groups to stick on proteins, and it gets them from ATP. So if Harmine is sitting in this pocket, then DYRK1A is unable to be active. So Harmine is a DYRK1A inhibitor. How do we know it makes beta cells replicate? Well, we showed that if you take Harmine and treat human islets with it, you can see increases in BRDU incorporation. That's a marker of proliferation. You can see increases in Cas67 proliferation. That's a marker that you all use clinically in insulinomas and pituitary tumors to see if they're benign or malignant. So these are proliferation markers that are widely used. And in contrast, if you treat them with no drug, human islets, there's no proliferation going on. So how much proliferation can you get? Oh, well, let me do this first. So what do we know about how it works? We know a lot about how it works now. And I'm not going to go through it. I just want you to see that DYRK1A is in the nucleus, and it serves as the brakes on beta cell proliferation. So the reason that adult beta cells don't replicate, or at least one of the reasons, is that they have active DYRK1A in the nucleus, and DYRK1A serves as the brakes. And so it makes sense that drugs that block DYRK1A would be able to drive proliferation. So DYRK1A inhibitors takes the brakes off, and now you're allowed to have beta cell proliferation. So where does this put us in the human beta cell proliferation story? So this is the proliferation we had when we were rate we had when we were 2 years old. Here's what it is with Harmine and human islets. So we can recapitulate that. That's pretty cool. But on the other hand, if I'm a type 2 diabetic and I'm proliferating at 1 or 2% per day, it's going to take me a long time to get up to normal. And if I'm a type 1 diabetic, it's going to take me a really, really, really long time to get back to normal at 1 or 2% per day. So a couple of years ago, I had a wonderful grad student join the lab named Courtney Akefi, and her project was to look for ways to get even higher rates of beta cell replication. So this is Courtney again working with Peng. So this was published last year. And what Courtney showed is that GLP-1 receptor agonists that you guys use every day synergize with DYRK1A inhibitors that I just described a moment ago, and they synergistically potentiate functional human beta cell regeneration. So what do I mean? These are the drugs we're talking about. These are the drugs that you all use. These have been on the market for a long time. We haven't studied DPP-4 inhibitors. I can talk about why that is at the end if you like, but what I'm going to show you should apply to them as well. These drugs are widely used around the world. 30 million people are taking them. They augment insulin secretion and they attenuate appetite. But none of these induce human beta cell replication. And people have looked at this for years. They don't make beta cells replicate. So here's the experiment that Courtney did that was so important. She was measuring Cas67 labeling in beta cells. Examples are shown over here. And she looked at 20 human islets from 20 different organ donors. Each of these lines is a different organ donor. And she showed that if you treat them with vehicle, this is just the diluent we use for Harmine, there's no proliferation. If she used plain old intestinal, natural GLP-1, nothing happened. If she used Harmine, as Peng had shown, at the same dose, 10 micromolar, now you see proliferation, and it averages somewhere around 2 or 3% in Courtney's studies. But if she combined GLP-1 and Harmine together, she now would see 7-8% proliferation. So this is an incredible rate of replication. This is the highest rate that anybody's ever seen. She also did dose response studies with GLP-1 and Harmine and basically showed that you get a nice, beautiful dose response. I mentioned that this response is synergistic. So this is a study that demonstrates that. So here's Harmine at 10 micromolar. It gives you 2% proliferation, which we're now calling 1.0. Here's GLP-1 by itself. It does nothing. And then here's a 10-fold lower dose, 1 micromolar Harmine. And so if you use 1 micromolar Harmine, not much happens to proliferation. If you use GLP-1 basically at any dose, not much happens. But if you add Harmine and GLP-1 together, now you get the same replication you get with a 10-fold higher dose of Harmine. So in classical pharmacologic terms, this is synergy. In practical terms, it means that you can use in humans a 10-fold lower dose of Harmine to get proliferation if you combine it with GLP-1 drugs. So that's where the synergy comes from. And that's super helpful to us in human studies going forward. So Harmine and GLP-1 synergize to induce unprecedented rates of human beta cell replication. Of course, then we wanted to look at the drugs that you guys all know: exenatide, liraglutide, lixisenatide, semaglutide, and none of them drive proliferation. That's no secret. That's well known. Here's Harmine again at 10 micromolar. But when Courtney added Harmine to any of the drugs that are currently on the market, she found that again she saw this dramatic synergy, 5 to 6% proliferation. So the message here is that any DYRK1A inhibitor, and we've looked at lots of them and it applies to all of them. So any DYRK1A inhibitor can synergize with any GLP-1 receptor agonist to induce these remarkable rates of human beta cell replication. So where are we now? So here's what we did when we were 2. Here's what we're doing now. This is what vehicle does, nothing. This is what Harmine does. And here's what Harmine plus GLP-1 receptor agonists do. In fact, it's actually higher than this, it's 6 to 8%. So how does that look for type 2 diabetes? It looks pretty good. 7%, 8% a day proliferation, that should get you back to normal. And what about type 1 diabetes? 7 or 8% per day, that also should get you back to normal. It would take longer because you're starting at a lower point, but it's going in the right direction. So we were excited about this stuff. And one thing that always has been a lingering annoyance for us is we're looking at Ki-67. And that is a great surrogate for beta cell replication, but it doesn't really prove unquestionably that beta cells are multiplying and you're getting daughter beta cells and granddaughter or maybe grandson beta cells. And so we wanted to measure beta cell mass over time. That's difficult with human islets because human islets only live in tissue culture for about a week and then they dedifferentiate or die. So you can't do long-term experiments. So we decided to look at this another way and ask if this proliferation is really real and if it works in vivo. So working with Adolfo, who many of you know, and Sarah Stanley, who many of you know in the Diabetes Institute, we took human islets, and we get these from NIH, and there are also commercial suppliers of human islets. We transplant them into mice with no immune system, so these are immunodeficient mice. We can put the islets with a catheter into the subcapsular space of the kidney, so you make a flank incision, pop out the kidney, make a little hole in the renal capsule, deposit the islets in there, withdraw the catheter, and now you can see the islets sitting under the kidney capsule. Then you pop the kidney back into the retroperitoneal space and put some staples in. And the islets are now very happy. They'll live for 6 months, 12 months, 2 years, the lifespan of the mouse in vivo. So this gives us an opportunity to study human beta cell mass and function over the long term. So now you can take these little mini implantable, they're called Alzette mini pumps, and you can load them with drugs, and they will last for a week, for a month. The ones that we use, they come in different sizes, but we use 4-week pumps, and we can infuse for 28 days, or we can remove the pump and put in a fresh one every month and get continuous infusion. So in the studies I'm about to show you, we went for 3 months. So we take human islets, put them into immunodeficient mice, and then treat them with vehicle, harmine, exenatide, or the combination of harmine plus exenatide for 3 months. At the end of the experiment, we harvest the kidneys, we fix them in formalin, and they're opaque, as you can imagine. But now there are tissue clearing techniques that are used in lots of areas of medicine and science. And Sarah's lab is doing them in brain. So we now can clear these kidneys, label them for insulin, they become transparent. And now you can use light sheet microscopy. Light sheet microscopy just takes a series of slices, Z-stacks, through whatever you happen to be imaging. So we basically can image the kidney like this, make millions of slices in it, and then you can basically load them all and generate movies. So here's what this looks like. So this is a movie of a mouse kidney. This is a mouse kidney. These little dots are glomeruli. The beta cells are the green parts. And what's cool about this is you can appreciate that, first of all, the beta cells are still there under the kidney capsule 3 months later. What you know intuitively, just like an MRI, this is volumetric. So not only can you see the beta cells, see the green, you actually know the volume of the beta cells, and computationally it's just generated and you have these numbers. So for the first time, we are able to actually assess and quantify in vivo human beta cell mass in a living system, in this case, mice. So with that, we then did the next obvious experiment, and this is Adolfo Garcia Ocaña. So if you take 300 islets, transplant them into the renal subcapsule of an immunodeficient mouse, and treat it for 3 months with vehicle, with Alzheimer's mini pumps, this is what beta cell mass looks like 3 months later. It looks pretty similar to what you started with 3 months ago. But if you do the same experiment and put in 300 islets from the same human islet donor and treat now for 3 months with harmine and exenatide, now there's a dramatic increase in beta cell mass. The fun part about this is, A, you're getting the quantification automatically since it's 3D, since it's voxels. But also, you don't have to know anything to see that there's a lot more beta cells here than there are here. So in a series of studies that Adolfo's done, and there are actually a lot more than this, but just to give you a sense of this, this is beta cell mass at the time beta cells are transplanted. In the first day after transplant, since the beta cells are ischemic, a lot of them die in the first 24 hours. And if you do nothing else, just treat them with vehicle and black, nothing, beta cell mass stays stable for the 3 months of the study. If you treat with exenatide in the gray, nothing happens either. If you treat with harmine alone, there actually is an increase in beta cell mass. It doesn't look like much on this scale, but actually it's about 2 or 3 times more than in the 2 controls. So harmine itself increases beta cell mass. But the clear winner of this beauty contest is harmine plus exenatide. The combination increases beta cell mass from 0.1 cubic millimeter to 0.7 cubic millimeters. So it's a 7-fold increase in human beta cell mass. So this is astonishing. No one's ever seen this before. Nobody thought this was possible. And without going into any detail, just say this is derived from a variety of factors. It's partly from proliferation. It's partly from enhanced function. I'll show you in a while that harmine also and exenatide make beta cell function better. They make more insulin. It also increases survival at the time of surgery. So there's this pro-survival effect. It's possible, but difficult to prove, that other islet cells may be transdifferentiating into beta cells. And we're trying to prove that. No one's got a good way to do that right now. And it's also intuitive from what I just told you that really a combination of these things are happening. So we're talking 7-fold increase in human beta cell mass in 3 months. So what does that look like in the setting of type 2 diabetes? If you increase this 7-fold from 1% to 7%, it would be way too much. This is too good for type 2 diabetes, assuming it translates fully to humans. You'd have to use lower doses for shorter durations, or maybe just use Harmine alone, because you wouldn't want to have a 7-fold increase in beta cell mass. In type 1 diabetes, as I mentioned earlier, it's not exactly clear where the starting point is in the average person with longstanding type 1, but let's say it's this. Let's say it's 0.1%, then you'd be up at 0.7% in 3 months. And if you kept going, you'd be normal in 6 months. If you're starting at 0.4, you might only have to go for 2 months to get back up to normal. Obviously, in type 1, you also need immunomodulatory therapies as well. This isn't going to fix type 1 unless something is done about autoimmunity. But this has simply never been an option before. So this is pretty exciting for us. So we worried next, okay, so now we have these great rates of beta cell replication. We're all MDs. We know that really rapidly replicating tumors are usually the most aggressive, dedifferentiated tumors. So we worried that making human beta cells replicate would make them dedifferentiate. So we did a series of studies, and again, this is Peng and Courtney, and asked, again, this is more than you want to know, but I just want to give you a flavor of it. So this is PDX1, and MAF-A and MAF-B and NKX6.1. These are transcription factors you must make in a beta cell to be a beta cell. This is GLUT2, it's the beta cell glucose transporter. So for example, we were worried that beta cells would dedifferentiate. If that happened, all of these things would go down. To our astonishment, we had hoped that they would stay normal. In fact, they actually go up. So you can see that they go up in basically in response to Harmine alone or Harmine plus GLP-1. We also showed that it happens at the protein level. So here's PDX1, MAF-A, and NKX6.1 in the nucleus of beta cells. They're beta cells because they stain for insulin. And the intensity of staining is much higher in the Harmine and GLP-1 groups than it is in the vehicle-treated groups. So these are essential beta cell transcription factors, and they don't disappear or diminish. In fact, they're accentuated in beta cells. So this is fantastic. This predicts that these beta cells are going to be super healthy, super functional. But these are all in normal beta cells, and we don't want to study beta cells here. We'd like to see what happens in type 2 diabetes. So Courtney, again, through NIH, obtained islets from organ donors with type 2 diabetes. And you can see that PDX goes up, MAF-A goes up, MAF-B goes up, NKX6.1 goes up, glucose transporter goes up. And so this is the PCSK1, by the way, is prohormone convertase. That's the enzyme that cleaves proinsulin into mature insulin. So this says that they're more differentiated. You can take type 2 diabetes beta cells that are dedifferentiated and redifferentiate them. And then this is a glucose tolerance test in dispersed islets. So here's type 2 islets with low glucose. This is about 50 milligrams per deciliter. Here's insulin secretion in response to 16.8 millimolar. That's 300 milligrams per deciliter of glucose. Type 2 islets don't respond very well to high glucose. But if you take type 2 islets and treat them with Harmine alone, that's this bar, or Harmine plus GLP-1, that's this bar, they have this dramatically improved response. So you can have it both ways. You could take beta cells and make them replicate, and not only do they not dedifferentiate, they enhance their differentiation and they function better than type 2. In fact, they function normally. We had never looked at proliferation in diabetic islets, and of course that would be important to prove. So using type 2 islets, Courtney also looked at whether the Harmine alone or Harmine GLP-1 combination could make human beta cells from donors with type 2 replicate, and the answer is it can, and you get the same kinds of replication rates in type 2 islets that you get from organ donors who are non-diabetic. So this is promising. I'm going to skip this slide in the interest of time and come back. All right, so everything I've shown you so far has been in islets transplanted into mice that were not diabetic. So it begs the question, can we really reverse diabetes for this? So this is work that Adolfo and Carolina Rossello in his lab has done, and recently this is just finished recently. So you can take immunodeficient mice, treat them with streptozotocin, and kill their beta cells and make them diabetic. When they're diabetic 3 days later, you can transplant human islets, and Adolfo puts them in both kidneys, some for iDisco mass measurements, some to look at proliferation markers, and then treats for 3 months with a series of LZ mini pumps. And then at the end of the experiment, at 12 weeks or 3 months, sacrifices them. He also measures blood glucose and insulin along the way. So here are the results of these studies. Here's day -3, the beginning of the experiment. They get streptozotocin on this day. They become diabetic rapidly with blood sugars around 350. If you do nothing more but put in a pump, and they're also transplanted with human islets, but a number of islets that's inadequate to fix their diabetes, 250 human islets. So if you do nothing else but put in a pump that delivers vehicle, they remain diabetic for 3 months. If you put in a pump that contains exenatide or a pump that contains a harmine, they get better, but they remain borderline diabetic. But if you treat them with a combination of harmine plus exenatide for 3 months, their blood sugar comes down rapidly and it stays down normally. It stays in the normal range for the entire 3 months of the study. This just says that these changes are statistically significant. But I want to show you also plasma insulin measurements. So plasma insulin is measured the day before sacrifice here. And this is the insulin. This is human insulin coming from these human islet grafts at the end of the study in animals getting a marginal mass of islets, 250 islets. If you treat with harmine and exenatide, it's about 4 times higher. So you can actually increase human insulin secretion or steady state insulin levels by 4-fold and reverse diabetes by treating with harmine and exenatide, in addition to enhancing beta cell mass. Why is it not advancing? Stuck. Oh. Slide doesn't want to go. Let's see. Where am I going? Slide. Let's try this. There we go. All right. So now on to a new area. So we know GLP-1 receptor agonists are safe. They're being used in millions of people. People would argue that there might be some safety issues, but from a sort of global use perspective, they're pretty safe and they're widely approved. So what do we know about harmine safety? Well, surprisingly, we know very little about it. We've done our own mouse toxicity studies, and I'll show you some of those in a moment. Other people have used harmine thinking that it might be good for memory or depression in rats. And the bottom line of this is that there are no adverse events in these studies reported by ourselves and others. We also know how much harmine it takes to kill a mouse or a rat, and it's a lot more than we're using in any of our studies or proposed studies. So what about human? Oh, sorry. This is some of our own safety data. So in these same studies where we infused harmine and exenatide for 3 months, at the end of the study, you can also harvest the organs in these mice. And so we harvested, again, we as Adolfo and his team, small intestine, spleen, heart, liver, adipose tissue, lung, pancreas, kidney, and brain, also not shown. That's a separate set of slides. And we harvested them from animals getting vehicle and animals getting harmine and exenatide. And we gave these in a blinded way to the veterinary pathology department at Mount Sinai. And what they found is basically no difference between the treated and vehicle-treated animals. So no tissue abnormalities. We also wanted to look at proliferation in other tissues, and that's all shown here by KS67. And just as an example, some tissues like liver don't proliferate much, but there's no difference in the harmine exenatide group. Other tissues like spleen have higher proliferation rates, and particularly intestinal crypts have high proliferation rates, but the harmine exenatide combination doesn't induce any further increase in the normal proliferation. So this is pretty good in terms of safety in animals. So what do we know about safety in humans? Well, harmine has been known about for a long time. It's got a reputation as being psychoactive. I'll tell you more about that in a moment. But these are the only 2 studies in the literature. They're from the 1950s, and they use hospitalized, chronically hospitalized psychiatric patients and gave them harmine orally to see what would happen. These are really bad studies that would never get through any IRB these days. There was no IRB then, and they therefore had no IRB approval. They had no consent. They didn't really tell you what the harmine that they gave is or how pure it was. They didn't measure harmine levels of blood. They couldn't measure harmine levels of blood. They didn't do any pharmacokinetics. They didn't tell you what an adverse event would look like if they found one. They didn't find any adverse events. Nobody died, if you believe the results. And the take-home messages from these, according to the authors, is that there were no adverse events at the highest oral dose, which was 960 milligrams. So hang on to that because I'll come back to that later. They also saw no beneficial effects. They were trying to use it for Parkinsonism associated with antipsychotic drugs, or maybe just depression or other sort of affective disorders. So basically, that's the entire world's literature on the safety of harmine in human beings. You may or may not have heard of ayahuasca. Ayahuasca is something that's used widely around the world, particularly in Latin America, but now has made its way to Brooklyn and Silicon Valley. And it contains harmine. It contains, as I'll tell you in a moment, it contains a lot of things. But basically, the message here is that harmine is actually a component of a street drug, ayahuasca. So what is ayahuasca? Well, in Latin America, you need to find this vine. It's called Banisteriopsis caapi. This is a vine, and you can chop it into pieces. Actually, the pieces that they use are these smaller pieces. This lives in the lowland jungle, moist, damp jungle in Latin America. And then you need to find this plant. This is called Psychotria viridis. It's a relative of the coffee plant, which is shown here. So this is a coffee plant. This is Psychotria. They look almost the same. And this grows in the shaded, dry mountainside. So they actually grow in different parts of tropical areas. So what do these things contain? Well, Banisteriopsis contains harmine and related harmaline and tetrahydroharmine. And these drugs are all monoamine oxidase inhibitors, or these compounds are. On their own, they're not psychoactive. They don't really have any psychological or psychoactive effects. Psychotria contains dimethyltryptamine. And dimethyltryptamine is a well-known hallucinogen. And so what the Mesoamericans found out is that if you collected these guys in the jungle and these in the shaded hillsides in other areas, you could mix these together and make this brew called ayahuasca, and you could get high. The biochemistry of this, or the pharmacology of this, is that dimethyltryptamine is a potent hallucinogen, but it's rapidly degraded in the GI tract and in the intestine specifically, and in the liver. So you can take it by mouth, but not much happens because its bioavailability is poor, and that's because it's degraded in the gut and liver by monoamine oxidase. It turns out that harmine, harmaline, and tetrahydroharmine are monoamine oxidase inhibitors, and they block degradation of orally ingested dimethyltryptamine. So basically, the combination allows you to absorb DMT, get a trip, and again, I love the fact that the Mesoamericans figured out that you had to put these two things together. So you can do this today. It's widely done. I went on the internet and asked how I could get an ayahuasca tour. So you can hire people to take you through the jungle to help you identify the plants. You can hire a shaman to chop up the Banisteriopsis, that's this stuff, into pieces that will fit into his pot. If you prefer, you could bring your own shaman from San Francisco to give you a more personal experience. You can take the leaves and the branches, throw them in a pot, cook them on a fire in the jungle to have the authentic experience. You can then filter it and make this beautiful ayahuasca infusion. And then you can have a solo personal party in the jungle by yourself, or you can bring your friends or your diabetes clinic to the jungle and do the same thing as a group. I'm being a little facetious, but when we reported that harmine could make beta cells replicate, I started getting emails on a daily basis from Latin America and from other people in the US saying, how can I sponsor trips to Latin America for ayahuasca tours to cure their diabetes? So what do we know about harmine and DMT in humans ingesting ayahuasca? Well, this group of investigators, I'll call them Callaway et al. From now on, went published in a journal that you may not read on a regular basis, the Journal of Ethnopharmacology, but they actually went to the jungle and found places where this is going on, enlisted volunteers and shamans to help them prepare it and administer this ayahuasca. And so they recruited a group of 14, let me get my pointer, seems to have disappeared. Hang on a second. They recruited a group of 14 volunteers. The shaman made the ayahuasca. The ayahuasca contained, they gave them 2 mils per kilogram, and that contained 250 milligrams of harmine on average in these 74 kilo people. So say 160 pounds or something. They also measured dimethyltryptamine, 35 milligrams, tetrahydroharmine, harmaline, and now harmine. So they gave that to the volunteers, and they took notes on their blood pressure and other things, their psychoactive experiences, but they also measured the pharmacokinetics of these drugs. So this is time 0. They've just drunk their ayahuasca, and then this is the curve for harmine. So harmine goes up to about 90 nanograms per mil at somewhere between 30 minutes and 2 hours. So that's the peak concentration of harmine, 90 nanograms per mil. And then it tails off. This is tetrahydroharmine. It's a breakdown product of harmine. So it appears as harmine gets degraded, and then it too is degraded. And then this is the curve of dimethyltryptamine. It also goes up because it's in ayahuasca, and because harmine as an MAO inhibitor is blocking its degradation. If you do this dose of dimethyltryptamine in the absence of MAO inhibitors, nothing appears in blood. So actually, this is the entire human experience with ayahuasca, or more specifically with harmine. And so this is if this is a representative ayahuasca preparation, this is what people are taking on a regular basis in really around the world. Remember that I told you that we had 3-month studies and that we were normalizing beta cell mass, normalizing beta cell function, reversing diabetes. So Adolfo also measured harmine so these are harmine levels in animals receiving harmine alone or harmine plus exenatide. And you can see the values are 4 to 6 nanograms per mil. So we're in the range where, this is how much harmine it takes in a steady state to fix, to give you a 7-fold increase in human beta cell mass and enhanced function. If you go back to this slide, this is what people are ingesting with ayahuasca, at least in this study. And this is the 4 nanogram per mil range or 6 nanogram per mil range. We're not even close. So the therapeutic doses in mice are nowhere near the doses that are routinely ingested in street drugs. So that's what we know about harmine in humans. So we think it's time for a phase 1 trial with pure harmine. We have 3-month toxicity data in mice. People's toxicity data says it's not likely to have adverse effects. It's in use normally in people on the street around the world as a recreational drug. So for lots of reasons, we ought to find out what sort of doses human beings can take. So it turns out that at Mount Sinai, James Murrow is a neuropsych clinical trial specialist. He's a psychiatrist who runs the GCRC, and he has worked with Dennis Charney over the years on FDA approval and commercialization of ketamine. So we have that. We've synthesized GMP quality harmine. We have a place that does that. We have it. We have gone to the FDA and said, can we do this? Initially, they said no, because we want to see that the stuff you make is what you say you can make. We've gone back to them recently with the results of our synthesis, which I could share if you're interested. And they've now said, go ahead. We have money to support this trial. And right now it's at the IRB. In fact, it was reviewed 2 days ago. We're waiting to hear what they had to say. But they already approved it in 2020, except to say that final approval will require hearing from the FDA. Now we've heard from the FDA, so we resubmitted it, and hopefully they'll tell us to go ahead. So we're planning to go ahead in the next few months. We think it'll probably start in late fall or January. This is what our formulation looks like, and we actually have made this stuff. It's in 100 milligram capsules that can be taken orally on the Psych GCRC. And then this is a letter to James from the FDA saying, let's go. This is basically, this is the key sentence. So you can go ahead and do this. So that's where we are with our own trial. We're excited about starting this soon. How does this fit in with other kinds of regenerative drugs or beta cell replacement therapies? So let's do some benchmarking. Well, right now, we do pancreas transplant. In the United States, there's somewhere around 1,200, 1,500 a year. Eyelid transplants have been done experimentally in the US. They're also done in Canada and around the world in lots of places. And again, globally, there are probably about, 500 or 1,000 eyelid transplants in the US. So let's say right now the volume of these is somewhere around 2,000 or 3,000 of these happening. It turns out pancreas transplant is very effective. It's very safe. The graft survival is very good. Eyelid transplant also works very nicely, not quite as nicely as pancreas transplant, but it's easier, less invasive. The bad news is that these aren't scalable. We're talking about millions of people with type 1 and hundreds of millions of people with type 2. You'll never be able to get enough organ donors to do this in millions of people. It's also very expensive. The cost per patient is prohibitive. You can't do millions and millions of people with these and expect to pay for it. And they also have adverse effects. Immunosuppression causes lymphoma in somewhere between 1% and 5% of people getting organ transplants. So it's not trivial. And there's also an association with other kinds of cancers, such as squamous carcinomas. Nonetheless, this is approved and in use by regulatory agencies. What about stem cells? This business of beta cell deficiency has been known for a long time. So let's take stem cells and turn them into beta cells. They can be induced pluripotent stem cells, or they could be human embryonic stem cells, depending on the country you're in and the regulatory bodies, et cetera. And you can turn them into beta cells. Well, I've given them a 3 on the Andy Stewart scale. The 4 is the best possible score. I gave them a 3 because they clearly do work well. You can get well-differentiated beta cells. You can transplant them into mice, and they work well. They're undergoing clinical trials in humans. I would say they're moving slowly, but maybe making some progress, but they're not approved for use in people yet. On the other hand, they're not scalable, right? I mean, we're not doing stem cell transplants in 900 million people around the world. We're probably not even doing it in 1 million people. We'd be lucky to do a few thousand people. And in part because it's work intense, and in part because it'd be extremely expensive when it finally comes to market. It also has its own set of adverse concerns. So, for example, the one people talk about is teratoma. My guess is that's not really a serious concern because the residual stem cells are removed by flow cytometry, but we don't know what the adverse effects will be in people until we really get into trials. In any case, it's not on the market. So if you look at regenerative drug therapies from this light, I gave myself a 3+ on the same scale because of the human efficacy data, 3-month, 700% increase in beta cell mass and reverse diabetes. That's pretty good. That's as good as stem cells. It's way more scalable than the other measures. The cost per patient will be trivial compared to these things. We don't know if it'll be oncogenic or not, and I'll say a word about that in a moment. Obviously, that's on our mind, but on the other hand, I've shown you 3-month clinical studies that don't show any evidence of that. Is it in use? Well, yeah, obviously GLP-1 family drugs are on the market. Harmine obviously is not. So what about mitogenic drugs and worries of cancer? Well, there are lots of mitogenic drugs on the market. Estrogens cause endometrial cancer, breast cancer. Testosterone causes prostate cancer. Growth hormones associated with colon cancer. Parathyroid hormones associated with osteosarcoma, at least in rats, although I believe not in humans at this point. And then, as Emily Gallagher will tell us all, insulin is associated, either endogenous or exogenous, is associated with increased risk of a variety of different kinds of cancer. So how do you deal with developing a drug and showing that it's not going to induce tumors? Well, obviously, in the cases of all of these, there had to be preclinical studies, extensive studies in pigs or primates or rats. or dogs. So we will have to do that. But then you figure out a dose and a duration. The thing that's exciting for me about this is, we use estrogens for years, testosterone for years, growth hormone for years, PTH for 2 years, insulin for years. We may only have to use Harmine for 3 months or 6 months and then be done. So yes, we're worried about oncogenic adverse effects. On the other hand, it's certainly worth moving forward to find out if they're there. So I'm going to leave you with a few conclusions. First, type 1 diabetes and type 2 diabetes result from inadequate numbers of insulin-producing beta cells. Obviously, type 2 is associated with insulin resistance as well. But if you have enough beta cells and can produce enough insulin, you don't get diabetes. It's funny, every once in a while. So almost all people with diabetes, even after 50 years of type 1, have residual beta cells, have some. So there's substrate there for regeneration. None of the currently available diabetes drugs drive beta cell regeneration or expansion. If you think about it, that's obvious. On the other hand, probably nobody really thinks about that too much, but that's clearly what's needed. And so what I've shown you is that residual beta cells can be induced to regenerate and do so at therapeutically relevant rates, retain differentiation, and it can be done with several different classes of small molecule drugs, meaning different DYRK1A inhibitors, which I haven't really gone into, and combinations with multiple different GLP-1 receptor agonists. So here's a few predictions. I think human beta cell regenerative therapy is clearly possible. I think that will happen. It's going to be effective in tens of millions of people with type 2 initially. My own guess is that it's more likely to go into type 2 because you don't have to deal with autoimmunity. And once it's established and on the market in people with type 2, and once immunological hurdles are overcome in type 1, then it'll move into type 1. We can talk about more of that more in the discussion section. It'll obviously be cheaper and more scalable than beta cell replacement strategies like eyelid transplant, pancreas transplant, or stem cell transplants. And it's going to be at least as safe as other regenerative therapies in current widespread use, and I gave you some examples of those. So what are the next challenges for us? We want to do this phase 1 human trial. We need to know how much Harmine we can give to people. So we just need to know that. We also need to develop more potent and selective DYRK1A inhibitors, and we're doing a lot of that. And again, in the question period, I can show you where we are with that. We need to do long-term preclinical safety studies in primates or pigs or dogs. The FDA will let us do a single dose study, but the FDA won't let us do a multi-week study unless we have, which we would need to assess efficacy, for example, in type 2. So before we can do that, we need to do large animal studies. So far, we've not seen any off-target effects. We see beta cell proliferation, but I've shown you we don't see effects in other organs. So if there are off-target effects, we'll have to identify a molecule to hitch our drugs to send to beta cells. But right now, it doesn't appear that we really need that. And then obviously for type 1, autoimmunity has to be managed. For type 2, that's not an issue. So in a sense, that might be a lower bar, easier target. So in closing, I want to tell you, again, I want to emphasize what Carol said, and this is for the fellows. I was a GP before I was a fellow. I wasn't planning to do basic science. I had no training in basic science. I was planning to go back into practice as a clinical endocrinologist. And then I did that for 35 years, the clinical endocrinology part. But the fun of this is you get to have it all. So this is a story that starts at the bedside. I ran the diabetes clinic at the VA at Yale for 20 years. So diabetes patient care, assessing clinical outcomes, which you all know are subpar, looking at complications happen relentlessly no matter what we do, trying to optimize therapy with every visit and never getting there in 70% of people. So then learning the basic science part, which I just want to emphasize, this is so learnable. Don't be intimidated by this. Many people in the room speak 6 languages. This is just learning different languages. So I decided, let's learn a little bit about cell cycle and beta cells. Let's learn how to do high throughput drug screens. Let's learn how to, once we discovered Harmine, figured out how it works and how to figure out what it is really, figure out how it works, figure out what we can do in animals as models and what we can do as medicinal chemistry. The animal stuff, MDs get in a second. You guys already know everything you need to know there. And then the med chem I didn't go into, but we've now made more potent versions of Harmine. And then here we go. It goes right back to people. Everything we've done that I've shown you is in human islets from organ donors. I'm an MD. Mice are super important. Rats are really important too. But if you want to fix human disease, you're going to have to deal with human tissue. So about 10 years ago, I just said enough of the mice. We're not doing that anymore. We're doing human stuff. Now we're going to phase 1 if that goes well. And if we can get the preclinical stuff, we can go to phase 2 and hopefully phase 3. And then what will happen? Then it goes to patients. It'll be good in some ways and it won't work in some ways. Then you go back to let's optimize it further. Let's find other partners. Let's find targeting molecules. So this is sort of the great Mandela of translational research. And again, for the trainees in the room, don't be intimidated by this stuff. I was less sophisticated than most of the people that we're interviewing today, for example, most of our current fellows. So this is the work of a lot of people. We have an insulinoma genomics genetics group that I won't talk about. The drug discovery part is here. We have how do DYRK1A inhibitors work, mechanism of action group. We have a human clinical trials group. So all of these groups work together, but I particularly want to acknowledge Peng Wang, Adolfo Garcia Ocaña, Courtney Akefi, Sarah Stanley, Bob DeVita, who's the med chem person behind this, and I haven't really shown you his stuff, Carolina and Alexandra, who've done the mouse model. They have taught us and actually done the mouse modeling that I've shown you. So let me stop there, and I'd be delighted to answer questions. I'm gonna stop sharing so I can see you, but I can put the slides up if people have questions. So questions.
So feel free to enter any comments. We already have a couple of positive comments about the talk or questions into the chat box. If you prefer to just unmute yourselves, go ahead and do so.
So Andy, that was magnificent. And I heartily endorse what you said, that this, doing research, I always liken it, if you can cook and follow a recipe, you can work in the lab. And what we bring as physician scientists is a knowledge of what the real needs are. And this is an ideal example of therapies based on what we know as the pathophysiology of type 2 diabetes in particular. If we could increase beta cells, we really could deal with the disease. I think it's really laudable that you're trying to do human studies. And as you're seeing, that's a long road to get to do those. Dogs are an excellent model. So, I hate the thought of anybody using dogs, but, there are some major diabetes in dogs investigators like Richard Bergman having thought, and particularly for mechanistic insights about starting there while you're waiting for the slow wheels of human investigation to process.
Thanks, Andrea. Again, I just want to emphasize the language part. If you're an MD, you speak your language. If you're a PhD, you speak a different language. Guess who has a more interesting life? People who can speak one language or people who speak 5 languages. If you speak Hungarian and Finnish and Gujarati and a little English, you're going to have a more interesting life than somebody who speaks only one of those. But they're only languages. So how do you learn languages? It's immersion. If you get dropped out of an airplane in Budapest, you're going to have a terrible first day. You're going to have a crummy first week, assuming you don't speak Hungarian. End of a month, you can find a bathroom, you can order stuff on a menu, and by 6 months, you can do everything you need to do. It's exactly like that in the lab. So the first week, you wish you were back in America or some English-speaking place where life is easy and you can get by with no effort. So you need to do the immersion, but once you do it, you can do stuff you never thought was possible. This high throughput robotic screen, as a fellow, I went to lectures at Yale. Like, really? I mean, do I really want to hear about, reporter cell lines? But actually, then it gets buried there. And, 20 years later, when we need it, let's do that. So don't be intimidated. If you're a trainee, don't be intimidated by the lab stuff. It's just another language. And one other part of that is, when I go to PhDs in the lab, say, okay, let's go start with human trials. They look at me like I'm insane. Like, no, you can't do this in people. Or like, yeah, you can do it in people. That's the whole point. We want to do this in people. So don't be intimidated by this stuff. I was a GP. I did a fellowship at Yale, and of course, in my interview, yes, I'm dying to do basic science. I've always wanted to do basic science. The truth is, I never wanted to do basic science. I never knew anything. Just had no interest. But as a fellow, I said, okay. I signed up for it. I said I'd do it, so I did it. I had a great mentor. I worked in a great lab, and okay, I can learn how to do this stuff. This is okay. And then things started working, and it's like, wow, this is cool. I think I want to do this another year, and then another year, and then another year, and then 10 years in, you know what? This is pretty fun. I think I'm in for the long term. So don't be intimidated by this stuff. Don't think you can't do it. You can.
Andy, it's Alice. I just want to say that the reason there are not a lot of questions is because that was a crystal clear talk. You can tell that you've written a lot of grants because you anticipate questions and you anticipate comments. So just the skill of giving a fantastic presentation was on display today. I also wanted to say, and for all of us, but particularly for the trainees, you show the utility of using the internet and PubMed. When you embark on a project, we're not the first people who ever thought of this. It may be something that's under a different name. You may have to be a little bit creative, but the fact that you dug up that article about the pharmacokinetics of ayahuasca, that saved you a lot of money, a lot of work. And it's good evidence that somebody else did this, and you can kind of go shopping. Well, maybe not in your own closet, but in other people's closet, and get a lot of things done, or at least get some clues as to it. So I think that that's really important for people going on research. The first thing you have to do is creatively look at what was done before. And for those of us who are a little older, this didn't used to be this easy. You used to have to go to this thing called Index Medicus. I mean, this is ancient history. So I think we all need to be aware of how fantastic this is, that it takes you about 5 minutes to go in and do a search. And sometimes a little bit longer if you have to look for a different name for something. So anyway, magnificent talk.
Thanks, Alice. I mean, again, I guess another thing, just along those lines, if people have questions, please chime in. I'm going to blather on if you like, if you don't ask a question, but.
So it looks like we do have a couple more specific questions in the chat box that are coming up. One from Dr. Bloomgarden regarding DYRK1A. He Googled that there's quite a bit of written about the relationships to its brain development, autism, possibly Down syndrome as well. Yeah. Can you speak to that at all?
Yeah, absolutely. So DYRK1A is on chromosome 21. And in Down syndrome, people have 3 copies of chromosome 21, trisomy 21, right? You may have all of three copies of all of chromosome 21, or just a part of it, or just this part, or just that part. But 100% of people with Down syndrome have 3 copies of 2 genes, one of which is DYRK1A. Okay, so if you don't have 3 copies of DYRK1A, you don't get Down syndrome. So overexpression of DYRK1A, and actually overexpression of DYRK1A is a bad thing for brain development. That's now been shown in mouse overexpression models and in Drosophila models. The Drosophila model of DYRK1A knockout is called mini brain, MNB, mini brain. So so DYRK1A is unquestionably involved in brain development. Too much DYRK1A is a bad thing. For that reason, DYRK1A inhibitors have been studied pretty extensively for a variety of neurodegenerative diseases like Alzheimer's, like Down syndrome, like other neurodegenerative diseases. And so there's a lot of study there. So that's why we went to such great lengths to look at other tissues, including brain. And I went through the synergy part of this quickly, but it's very important. The fact that we can give this together with GLP-1 means we can use 10-fold lower doses of Harmine than we need to see effects in other tissues. So GLP-1 is clearly enhancing proliferation, but it's also giving us a beta cell targeting opportunity that we wouldn't have if we used pure Harmine. So bottom line is, some people are developing DYRK1A inhibitors as neurocognitive improving drugs. People are also looking at them as anti-cancer drugs because DYRK1A is frequently upregulated in cancer. The biology there is you're giving chemotherapy, it's killing the tumor. Now the tumor gets smart, starts overexpressing DYRK1A and turns off proliferation. Now your anti-proliferative chemotherapy drugs don't work. Let's give them a little DYRK1A inhibitor to get them proliferating again, and now we can kill them with chemo. So it is being used in other tissues, in other contexts being developed. And again, ultimately it comes down to safety studies. I guess the last thing to say there, and I didn't talk about this, but Bob DeVita, who's at Sinai now, but was the director of MedChem at Merck for many years, has now made hundreds of versions of DYRK1A inhibitors that are 30 to 100 times more potent and more specific. So if Harmine isn't the drug, and Harmine probably won't be the ultimate drug, we've got a whole library of additional DYRK1A inhibitors that we're characterizing that may be a bit more effective.
It's fascinating that the beta cell has so many similarities to neurons and all the various growth factors and so on. So this really fits into that story.
Yeah, you're right about that. I mean, so like, in the world of GWAS studies, so we get all these hits, and if you're a beta cell person, there are beta cell hits. But if you're a brain person, hypothalamic controlled metabolism person, they're all brain hits. Because all the GWAS hits, many of the GWAS hits are expressed in both. And again, for the fellows, it just illustrates how little we know. If you think that everybody is smart upstairs, who's already smart and knows everything, my view on it is none of us know anything. There's so much more to learn.
So we're a few minutes over time, but if you have time, we have a couple more specific questions for anyone who'd like to stay on. So one question about the potential therapeutic applications for Harmine, which are, if you can speak to this, which are possible based on your research, and how was the name derived?
The name comes, so there's a plant in the Middle East called harmal. It's also called Syrian rue, R-U-E. And so it was extracted, I should say plants in the Middle East of the Paganum family also contain Harmine, and they also contain other things, but then they also have psychoactive activity. So again, from Fertile Crescent era, people have been using extracts in the Middle East that contain Harmine. So it was, I guess, initially identified in Syrian rue, which in Arabic I understand is called harmal, and so Harmine comes from harmal. That's where the name comes from. What was the first part of the question? Oh, other uses or other next steps?
Therapeutic applications, yeah. Mm-hmm.
You mean beyond diabetes? You mean?
That's my guess. Yeah, based on the question.
Yeah, I mean, so people, as I mentioned, people are developing DYRK1A inhibitors as anti-cancer drugs. They're also developing them as anti-neurodegenerative drugs. And again, there's an opportunity here. If you need to do targeting or making it specific for a particular tissue, there's a lot of designer medicinal chemistry, and that's what Bob DeVita does. So we may need to figure out a way that targets it exclusively to beta cells, or we may be able to make versions that work exclusively in beta cells and not other tissues. That's where this is going now.
Right. And then we'll finish up with the final question from Dr. Rayfield. Any hypoglycemia that you observed in your studies with Harmine and exenatide, and whether glucagon levels are measured, and what were they?
Yeah, thanks, Elliot. That's a really good question. And the answer is no. And we've looked hard. When I showed you those long-term studies with blood glucose, we look for hypoglycemia. There's no fasting hypoglycemia. So it looks like we're regenerating functional beta cells that know when to stop making insulin. Yeah.
That's great. Thank you for that. Excellent talk.
Thanks, Elliot.
Great. So I think we'll wrap up there. Thank you so much.